What's up, guys? It's Mr. Schmidt, and here I am to share with you guys section 15.2. We're talking about human genome disorders. Just let it cook. Let it cook. Don't forget to like and subscribe. Yep, it's going to be great. I've got a bunch of followers. I've got over 100 followers. Anyways, check it. I have a lot. I've had my YouTube for a very long time. So, human genetic disorders. When we talk about genetic disorders, as we mentioned earlier, we're looking at proteins and protein formation and the problems that occur if you have extra or too little genetic information. Two main lecture lesson points we have here. They are the effects of meiosis. When you go through meiosis, if you have a non-disjunction event, you can wind up with an incorrect number of chromosomes in the gametes. The other one that we got going on, small changes in DNA can make big changes in the traits of a person. Little changes can make big changes. What's up, guys? What's up, brother? Let them cook. Okay, here we go. First piece of information, non-disjunction. During the process of meiosis, your cells go through 2 rounds of cell division, mitosis. 2 rounds of mitosis, I should say. It's not really cell division, it's mitosis. And as they go through those 2 rounds, we take the genetic information that we have and we split it. So you see here, this is when we have our 2 tetrads. In a normal human cell, there would have been 46 copies of, or duplicates, or doubles of. To simplify the drawing, we don't have that many. We just use a couple of them, just so we can get an idea of what we're trying to do here. And so as we go through non-disjunction, when we have this non-disjunction during meiosis 1, what that means is we're supposed to be splitting these in half so that this set of chromosomes goes to 1 and this set of chromosomes goes to 1. Well, if that split doesn't happen, you wind up over here an extra pair of these chromosomes. And over here, you're missing a pair of chromosomes. I wonder if the mouse is going to show up. I have no idea. So with this non-disjunction, when we get all the way down to our gametes, you're going to wind up with 4 gametes that have the wrong number of chromosomes. This is why a non-disjunction event during meiosis 1 is a little more detrimental, a little more problematic than non-disjunction during meiosis 2. Because meiosis 2, as we talked about in the interactive, you still will wind up with 2 that are okay, but the other 2 would be bad. Here, all 4 are bad. Okay? Gametes with any abnormal number of chromosomes may result in these gametes here, when they form, like if this is a sperm cell, the sperm cell could join with an egg cell, and that individual will have 1 too many, whatever chromosome this is. It might be an 18, it might be a 21, it might be an X, who knows? Okay. Continuing, Down syndrome. Down syndrome is one of the most common trisomies. Show of hands, how many of you have met someone with Down syndrome? Okay. There's actually a student at Trinity who has Down syndrome, Jojo. And Jojo has 3 chromosome 21s. And as you probably noticed, he also has a particular set of facial features, a particular set of understanding. There's a slight amount of mental retardation, not quite full capacity. Okay, yes?
Is there a reason why it's more common on chromosome 21 than other?
Not specifically. I can tell you that it's mildly more common in late life pregnancies, so like after 40 years old, because, and we'll talk about it more in anatomy physiology, but when the eggs are in stasis, they're kind of like waiting to be fertilized for 40 years. It causes them to kind of not really get sticky, but they don't divide quite right towards the end of that time. So like when you're super old, not super old, but when you're older and have kids, there's a higher instance of these genetic deformities happening. Yes?
Does that happen if like you already had kids and then it's so late?
So if you have like at conception, the blueprints for that person are developed. So it's not like now during, when you turn 16, you suddenly pick up another chromosome. It has to happen at the very, 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 very beginning.
You said like how the eggs are like waiting or something.
Oh, yeah. So, for example, when you're in your mom's womb, the eggs as a female, as a daughter that you're going to have are already developed and developing. And they will sit there kind of waiting inside your ovaries until the time comes where once a month, roughly, they'll be released through the menstrual cycle. And again, we'll talk more about that in bigger detail in the NatFiz. But those eggs then, from shortly after conception, like when you as an infant inside your mom's womb starts making eggs, they sit waiting until they get used, and it could be 40 years. And so when that happens, this meiosis process, typically it's meiosis 2, what happens is they go through meiosis 1 and then they wait, and they do meiosis 2 right before the ovulation occurs, right before they're released. And so they just sit and literally just wait. And so if you have kids when you're 18, 20, 25, those eggs have not been sitting around as long as eggs if you wait till you're 40, 45, 50 to start having kids. That kind of makes sense? Now, as far as chromosome 21, I'm not 100% sure of why like 21 and 18 and 13 are kind of the more common ones. I think it might be because if you're trisomy something else, the baby doesn't make it to full term. The baby might miscarriage inside the womb because some of these chromosomal deformities might not even allow the person to survive. Okay, good questions. Yep.
Is Down syndrome only found in like the chromosome 21?
Yes, that's why it's linked to this trisomy 21.
What would be like the trisomy?
You can do trisomy 18 is another example.
Like any of them?
It can, however, we only see it in viable people that live, like trisomy 13, trisomy 18, trisomy. Because if it was trisomy 1, there might be too much information here for the person to survive. So they wouldn't even get born. They would actually die. They'll like be miscarried by the mom's womb.
If it's in like a miscarriage, is that like a way of like protecting it?
Correct.
Like the mom
Yeah.
too?
And I like to think of it like that's kind of God's insurance policy, if you will, of like this baby's better off with me up in heaven because they're going to have too many struggles here on earth. So that's kind of how I look at miscarriages. But okay. All right. Other disorders. And there's a couple of them. If you kind of Google them up, you can see them. And at the end of the lecture here, I can probably bring some images up if you like. But this example here, Turner syndrome, this would be a person who only has an X chromosome. So the example we saw in our interactive had a karyotype where there was just one X. In Turner syndrome, there are a couple of facial features that are different. The individual would look like a female, but wouldn't have the full reproductive capabilities of a female. And the wild thing is it's in all cells. So every single cell is going to be missing that one X chromosome. Because at the very beginning, at conception, that individual's genomic structure was set, and it hasn't. You can't add another X chromosome in later.
So are they still female?
So they will look female. Yep, they will have the physical characteristics of a female, but their development may be different. Okay. Klinefelter syndrome. This is another example, non-disjunction. So a person with Klinefelter's would have XXY for their traits. And so they would look male as far as reproductive structures, but they would also have kind of like breasts, and they would kind of have just some female characteristics as well that would kind of be mixed in there. So those are some of the characteristics that you would see with a person with Klinefelter's. So a difference in the number of chromosomes can have vast effects on the development of a person. Good on these two? Good. Yeah, I think my one wire here is getting a little bad. So, all right, from molecule to phenotype, as we look at the different types of amino acid strands or strings, when we get changes in the DNA, that's where we're going to wind up having changes in that amino acid sequence. The example shown here is what's called sickle cell disease or sickle cell anemia. Sickle cells are cells that are not shaped like a standard red blood cells. Normal red blood cells are kind of round. They look sort of like an inner tube. We talked about, if you go down a sledding hill, you don't ride down on a curved, slicey thing like this. You ride down on a nice round tube. That's because they flow well through that situation. Your blood cells also, as a nice round structure, flow well through your blood vessels and through your capillaries. Sickle cells, however, look like a sickle. So if you've ever seen like the Grim Reaper, that cutting blade that you see is kind of like half moon shape. That's called a sickle. These are the shapes of these cells. They kind of have this curve to them. That shape does not travel well through blood vessels. In fact, it gets jammed up and causes traffic jams that then cause swelling to occur in that area, pain, discomfort. And so that small mutation, and all it really is one mutation that causes a protein that normally is round to go straight. So inside here, there's like straight strands of a particular protein that stretches out the blood cell, making it kind of form this shape. So differences in amino acid sequences can cause differences in phenotypes, and those show up in different diseases. Another one that we looked at in the interactive was cystic fibrosis. This is from the deletion of 3 bases. So if we take a CTT out of the normal cystic fibrosis protein, we get this cystic fibrosis, which you can see this person here is being treated for cystic fibrosis. It basically produces a very thick mucus in your lungs, making it really hard to breathe. And it's not something that you can just take a medicine to cure. You can take medicines and things to help alleviate the symptoms. They loosen the mucus up, allow you to cough it out, spit it out a little better. But it's kind of like having a chest cold all the time. Not fun. And as I mentioned, it's genetic. It's not like you can just make it go away. It is permanent. Yes?
If you don't have it, like, can you like
Develop it?
yeah, develop it?
Not really. One of the other students from the last class asked about like asthma and how asthma can kind of develop. Asthma is a similar kind of scenario where your lungs have this reaction.
Our Fayed, you will go to room 6. Please bring your iPad. And would Emmy Troop and Ingrid please come to the office?
They have a reaction to the atmosphere, and it causes.
And now for your prom court
Okay,
representatives.
I guess we're going to pause
For
here.
the senior class, your representatives will be